Good day, this is the Professor, CBMD PhD, and today's Monday, November 6th, 2023. It is 3:10 and 26 seconds, a.m. Eastern Standard Time, and today's research, cancer and Telomouse. So let's jump to it here. As scientists create special Telomouse with human-like telomeres, date October 31st, 2023. Source: The Hebrew University of Jerusalem. Researchers introduced the Telomouse by making the subtle genetic alteration in standard laboratory mice. They made the mouse telomeres, which protect the chromosome ends, more closely resemble those in humans. The Telomouse model developed by incorporating a genetic variation from a mouse species with naturally shorter telomeres provides a valuable resource for in-depth aging and cancer research. The discovery promises to reveal new insights into the genetics of aging and may contribute to enhanced longevity and well-being, as telomeres hold a pivotal responsibility in safeguarding our genetic material and ensuring the orderly division of our cells. Maintaining their structural integrity and optimal life holds the potential to diminish the risk of cancer and facilitate a healthier aging process. However, a significant hurdle has emerged. Conventional laboratory mice possesses telomeres approximately 5 times longer than those in humans. This disparity has posed a formidable challenge in using mice models for comprehending the implications of telomeres for human aging and cancer. In the development of the Telomouse model, researchers turned their attention to a distinct mouse species, M. spritus, notable for its inherently shorter telomeres within the genetic code of these mice. A subtle variation within a pivotal protein known as RTEL1 was identified, and by transferring this genetic distinction into typical laboratory mice, they succeeded in producing a lineage of mice with human-like telomeres. These telomice exhibit robust health and reproductive capabilities, making them an exceptional resource for in-depth investigations into the complex realms of aging and cancer. And this study illuminates the central role of RTEL1 as the arbiter of telomere length. A nuanced modification to this crucial protein has enabled scientists to fashion a mouse model that closely approximates the human telomere length. And during the researchers also achieved an invaluable breakthrough in their ability to measure the length of each single telomere, and particularly the shortest telomeres in the cell, which are the ones that dictate the cellular function and fate. The scientific investigators and microbiologists developed a method for measuring the precise length of individual telomeres using a new generation of DNA sequencing called nanopore sequencing. And this method termed nanotelsec enables evaluating the telomeric health in samples from blood or other tissues in healthy individuals, as well as patients of cancers and aging diseases, and improve diagnosis, prognosis, and treatment of these patients. The Telomouse model is promising to enrich our comprehension of the intricate nexus between telomeres, cancer, and the aging process. They believe that nanotelsec will replace currently used methods, enable accurate evaluation of the telomere state in patients and healthy individuals, and reveal how it affects human health. Such insights will hopefully culminate innovative strategies for combating cancer and fostering the well-being of aging individuals. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Teacher of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.